Hello and welcome to this episode of Let's Talk, where we continue the conversation that belongs to our very interesting and very scientific session titled Peri-Implant Diseases, Immunoresponse, Cellular Crosstalk and the Microbiome. Well, let's say what we can say about that. I'm now joined by our three expert speakers. Tobias Fretwurst, you're from the University of Freiburg in Germany. Welcome.
Thanks.
Next to you, Daniel Jönsson, Associate Professor at the University of Lund and Malmö in Sweden. Welcome, Daniel.
Thank you.
And next to you is Asaf Wilensky, Professor at the Hebrew University, the Hadassah School of Dental Medicine in Israel. Asaf, welcome.
Nice to be here.
Let's talk a little bit about this very interesting session, whether you've watched already the full recording of the session and we continue the conversation or we tease you into it. Let's start with the basic. Peri-Implantitis. If I ask you now, what is the incidence that we currently lean on?
Yeah, so if we're looking into the literature, it's around about 20% incidence. It's the studies of Jan Derks from Sweden. There are systematic reviews on that. And I think that is pretty high, actually. What we have to keep in mind is that we have really good survival rates in implants. So always if we hear we have a high incidence, we have high survival rates for 10 or 20 years.
Yeah, so you're saying the disease might occur, but it might not result in an extraction or a failed implant.
Yep.
Daniel, Asaf, when I Google, I see 10 to 50%. What do those figures mean?
10 to 50% mean that you have some kind of bone loss around the implants. But as Tobias just pointed out, that doesn't mean that the implant needs to be extracted. It needs to be treated and it needs to be followed up. But it can hopefully survive for many more years.
Yeah, you showed us some studies on the success of treatment. What are the figures there?
In a 5-year period, the success of peri-implant treatment is pretty low, unfortunately. It's around 50 to 60%. But as I also show in the presentation, we have peri-implantitis and we have peri-implantitis, and you have to kind of be pragmatic in how you treat those patients. In one instance, you may have kind of the cutoff of where we consider it to be peri-implantitis or not, and the treatment in those patients are pretty successful, and the numbers would be higher if we only included them. But then we have patients that are older and have had implants for a longer time that have lots of bone loss around their implants, and where you have to be pragmatic. And if you want to have good numbers, well, then you can extract all the implants, do huge bone augmentations, and put in new implants. But in some of our elderly patients, that's not a good option. So then we continue to treat these implants and lower the inflammatory burden and have the implants and have them survive even though they are not considered to be successful peri-implantitis treatments.
Exactly. So we really need to look at the specifics of the case. I think that was a message that was in all three of your talks. Asaf, peri-implantitis seems to be the big mystery in the field of implant dentistry. What fascinates you about this topic?
What fascinates me is it's a multifactorial disease. We don't know a lot about it. We don't know how to treat it with a very good prognosis. So the challenges in peri-implantitis are huge, and it's nice to have a research focused on a disease that no one knows a lot about it. So these are the challenges that I'm fascinated.
Yeah. And in this session that belongs to this talk, all three of you shared extensive science on your explorations on what it could be. Tobias, you introduced us to the coffee machine. Could you quickly tell us the key message from that analogy?
Yeah. Thanks for giving me the time to point on the coffee machines. So this is an analogy, an image I just created for the talk, is that the coffee machines are different if we're comparing them on a microbiome level. That isn't a silly study. It was not a fun study. They wanted to know which bacteria are on coffee machines and how can we maybe do anti-infective therapies, or how effective is the coffeine in the coffee beans. And I transferred this picture to our peri-implantitis patients. So we see different microbiomes, so the entirety of the bacterial species from patient to patient. And then I transferred it to the next level on the immunome, on the immunological cells, and even on the proteome, on the protein cells. And so that was my claim, that we see each patient as an individual. We do that, but also in terms of the peri-implant disease immunological reaction.
Yeah, and a bit of a spoiler, you looked at all these 3 areas, and of all you kind of concluded, well, each patient is different. There's no conclusive indicator of where we could base future therapies on.
100%. What we found out on our first material we have now, and we need big data, maybe we discuss this later on, we find some pattern in the proteome level, so on the protein level in the clavicular fluid. There might be a sign we can use, but still we are on the first step. You're right, yeah.
Then, very interestingly, Daniel, in your talk, which was second, you also kind of built on this. You showed us more about the microbiome, the potential options. You came to similar conclusions. Everyone is different and everything is different. Yet your conclusion at the end of the talk was, I still think it's a pocket of microbiome. Prove me wrong. What made you make such a strong statement in the end?
So what I really bring forward is that when we look at the peri-implantitis microbiota in all of these studies, in this numerous systemic reviews that have been done on this, the top candidates of the most important species in the peri-implant microbiota have previously and are still very much associated with peri-implantitis, with periodontitis as well.
Yeah.
So it's interesting to see that even though there are some bacteria that stick out, both in peri-implantitis and also in periodontitis, the bulk of them and the vast majority of the high and most significant
families,
species, and the ones that are most highly associated with the different diseases are the same.
Yeah.
So it seems that there's a common pocket microbiota, and then there are some a little bit more specific to peri-implantitis and some a little bit more specific to periodontitis, but there's a bulk that's common in both diseases.
Tobias, I found that difficult as an audience member, because I think, didn't you just say all coffee machines are different, so we cannot generalize like Daniel was doing?
Yeah, we are actually on the same page. Good question, good question. We are actually on the same page because what I showed in the talk, and that's why I looked the talk, that if we obtain the samples from different sides of the coffee machines, we see different microbiota. Yeah. And that is exactly what you are claiming. If we're looking in the pocket, we will see different microbiota than in the oral cavity or in the saliva or maybe in the gut. What I find interesting as well, now we are looking just with species are there. The next step would be how they are connected. Yeah. So like network, and we are nodding. So which are the network we see, what is the virulence, and there are new studies from Japanese groups, from the Tokyo Dental Medical University and so on, which show we don't have to show the bacteria there, we have done that. Now we have to show how they are interacting with each other, and you make the good point, Daniel, and then how they are interacting with the immune system.
Yeah, exactly. Those of you hoping to hear some clinical advice, stay tuned. It might be difficult because we were deeply exploring. Asaf, you presented results from an experimental study in mice.
Right.
Very interesting findings. Let's again start with the basics. Why is it either easier, or what's the biggest challenge in translating findings in mice to perhaps situations in humans?
I think the big difference is mice are the same. When we are exploring or investigating mice, they all have the same background. With people, as Tobias said, we have a big variety. The immune system is different in humans. The microbiome is different with humans, which is not the case in mice. So it's easier to start your research with mice and then translate it to humans.
Maybe I can add here, Asaf, because if I think most of the audience are not doing animal research. And so I did that in Harvard and Michigan. And so if we are getting rodents like rats or mice, they are the same strain. So they have the same genetic background. And that is what Asaf is saying. You order always almost the same individual. And that's why we have a problem to transfer this data to our patient, where every patient has different genetics, different microbiota, and so on.
Because mice are like identical twins.
Exactly.
Yeah, perfect.
And they eat the exact same thing as well.
Yeah.
Exactly. But then, so it's not even the challenge that mice genetically or physically are so much different from a human. It's the fact that your study is kind of in a homogeneous group. That's
right. Exactly. Yeah.
Next thing then, talking about similar results, what was fascinating, and I also saw that coming in as an audience question, it seemed that in the results you showed, all the mice had to deal with bone loss around their microimplants. Right. Does that mean that you saw peri-implantitis in all study subjects?
In mice, I saw it.
In mice, yes.
Yeah. All mice showed gradual.
How do you explain it? Because we just learned that in humans, peri-implantitis has a high incidence, but not everyone is receiving it.
So first, the immune response is different. The microbiome is different. As I said before, so people and mice are completely different. And if we wait enough time in humans, maybe we'll see peri-implantitis in all of humans. So I'm not saying it will happen, but if you wait 20, 30, 40 years, maybe you'll see peri-implantitis in all humans. Anyway, in mice, this is what we found, and mice are different than humans. We should keep it in mind.
Yeah, true. But I'm still fascinated by the fact that in all your subjects, bone loss was happening.
Yeah.
Do you understand why?
As a group, statistically, we found... Yeah.
Okay.
Okay.
Tell us a little bit about a resolution, perhaps a hint for a treatment option in the future. You introduced, I believe, a substance called Resolvent. What is that?
Resolvent D2 is lipid mediators of inflammation. These mediators are naturally produced by our body, and they have the ability to actively terminate inflammation. Because in our body, the inflammation doesn't go away. There are some mechanisms that stops inflammation. So we use these mediators in order to modulate the immune response around implants. Because we don't think that mechanical or antibiotic treatment, at least in mice, helping resolve peri-implantitis. So we focused about modulating the immune response.
And you showed amazing results. Daniel, Tobias, do we think this is now enough to see if we can reproduce these kind of interventions in humans?
I think that Resolvents are very promising. And as you mentioned, they are sort of the cleaning up after the neutrophils and everything that they have done in the tissues. And as you mentioned in your talk, peri-implantitis is much more related, or what? I don't remember who talked about it, but
it was a big talk.
It was a big talk.
90 minutes.
But that neutrophils are so important in peri-implantitis, and so if Resolvents will have a big impact on something, I mean, periodontitis or peri-implantitis, peri-implantitis is probably even more important than periodontitis.
Yeah, but I think we shouldn't focus on one treatment modality. So we will not find the golden bullet, in my opinion, because of the differences between our patients. I think that is the step in the right direction to use maybe anti-inflammatory substances like this, or you showed other ones. Reason we did some studies about that. They are shifting certain immune cells from more inflammation to more silent inflammation or to anti-inflammation. But yeah, every patient brings a different immune system. And now we are starting to influence this immune system. And now it will be getting really interesting, but dangerous, or chances are there. And we need techniques to monitor that. If we're altering that, We need to monitor that. That can be species, so bacteria. It can be proteins, immune cells, and that's what I claimed in my talk, and we're all on that page. So to be more precise in our decision and to monitor that. So precision implant dentistry. Yeah.
Precision implant dentistry.
Yeah.
That's an interesting term. As a clinician, I now think, okay, I need to navigate more securely. No, that's not what you're talking about. What do you mean with that term?
Yeah. So so we are quite precise in dentistry. Otherwise, it would not work. It's a term, precision medicine. It's coined 10 or 20 years ago in Stanford, Leroy Hood and other people, that we need biomarkers or any factors we can measure to see how our therapies are going and how we can monitor that. So it's preventive, it's precise, and then there are one or two more terms they are using to get more information out of our patients. And we use what we are doing, metagenomics, immunome data, proteomics, and they are gathering biomarkers, and these biomarkers we can use to create better therapies. Yeah.
Daniel, what does that look like in the future? What does the clinician do? Do I first have to provide a sample, blood sample, saliva sample, and you first analyze and then decide my treatment protocol? Or where does it?
Maybe. I mean, I really agree with that. That's how biomarkers should be used to really see...
Kind of as a predictor.
As a predictor, and to see what type of patient do I have in front of me? Why do we have this situation that we have ended up in? I mean, much of the research between peri-implantitis and periodontal disease had been sort of standing still because you can't... People say that, well, this doesn't explain all the cases of periodontitis. This doesn't explain all the cases of peri-implantitis. We're never going to get that. We need biomarkers to sort out the different groups of people that we have that may have these diseases for different reasons. Okay. Did that answer your question?
Yeah, a little bit, but it explains the feeling that you indeed get in the session. Because, again, it's impressive scientific work that you present, super interesting, but you also kind of close certain pathways. So it's probably not that, it's probably not that. It might be a little bit of everything. So it also leaves us a little bit, now what? Asaf, now what? What is the next step? Let's take it from a scientific perspective. What would you like to explore further now with the findings?
So we're just about to finish a big project comparing titanium to zirconia. Because as I've shown...
Diving into the aspect of material choice.
Right. Because if titanium is not the ideal material, maybe we should look for a different one. So we dived into the difference between zirconia and titanium, as Tobias showed us in his lecture. And we found very promising and nice results showing the differences in the immune response around zirconia and titanium. So maybe one lead could be a different material, or maybe some other genetic work with teeth buds, or I don't know what's the future.
And Gerrit, I think it's another point, because you pointed out, okay, we don't know what is this, what is that, everything is so wishy-washy, we call in Germany or in Netherlands as well, so not precise enough. And that is what Daniel brought up, and I tried to bring up in my talk as well. We need the big data. It's not enough to look into one coffee machine or one patient, or this really interesting probiotic systematic review was 85 patients or 90 were included in that. It's almost nothing. We need data for 1,000, 10,000, 30,000 or millions of people to get out of our high sophisticating analysis method good data out of it. Yeah. And then we see pattern, and we will see that because we see this in aging research, in epigenetic pattern, and we will find this in peri-implantitis as well.
Yeah. We need to work together for big data.
Yeah.
Daniel agrees. I can tell you, it's not the first time in this Let's Talk studio that this is being said. Question remains, okay, we need more data. How and where? I mean, who's in the lead? Where do we get this from?
The way to do that is to work together, to not be too protective with your data, to share. Labs all over the world sharing data.
Currently, that could be done more. I'll phrase it subtly.
Yes, definitely. And I mean, working together, sharing data, sharing samples so that we can all, I mean, when it comes to proteomics, microbiomics, metabolomics, everything, you need to analyze them simultaneously to remove all sort of errors. And to do that, you need to have all the samples at the same time, at the same place, and bring it together and work together, analyze together, and then we can come up with something more.
Beautiful call for action, Asaf.
We need to collaborate more. We just talked to Tobias and Daniel about collaboration sessions.
Coming out of this session.
Yeah, sessions like this have great opportunities.
Beautifully. So I guess that should be the clinical impact then. If any clinician is watching and they see an opportunity to enter or to submit data to a scientific project, please sign up, take the work and collect the samples. And I think that's a nice tie into another session. First of all, I invite you to make sure you check out the full recording of what these gentlemen have presented on stage. But it's also a nice tie-in into another Let's Talk we had here in the studio about plenary session 1, where we talked all about the potential of big data and also analyzing that with artificial intelligence and so on. So I think with gentlemen like the 3 of you here with us in this Congress and in your respective research programs, and you all coming together and providing all the data, I think there is a bright future in front of us. Thank you all for being here. Thank you for sharing, and see you in the next video.